So first, thanks for organizing this. I just wanted to, so I'm a postdoc with in David Goldstein's group, and I want to talk about some work that we've been working on regarding looking at transcriptional inhibition as a strategy for basically knocking down host proteins that are essential for virus entry into the cell. And I just also wanted to note that the work that I'm presenting right now, we just put it on as a preprint. The link is over here. So if anyone wants to read this in more detail. So I just have one slide of introduction where basically there's a lot of work that's been done on the ACE2 receptor, which is essential for the virus to enter the cell. But we looked at also other host proteins that are essential for viral entry into the cell. So in addition to the ACE2, there's also TMPRSS2, which is a host protease that's involved in sort of cleaving the spike protein and facilitating entry. And in addition, there's also cathepsins B and L. In terms of essentiality, ACE2 and TMPRSS2 are both essential for viral entry, while cathepsin B and L are dispensable. So we mostly focused on ACE and TMPRSS2. We considered both of these genes as potential targets for transcriptional inhibition. And it seems like ACE2, at least in terms of looking at knockdown, is not a great target, mostly because in the human populations, its loss of function of ACE2 is really strongly selected against. So there's it's highly intolerant if you have a loss of function in ACE2. And also in studies of SARS CoV-1, it seems like ACE2 itself seems to be protective against lung injury. And so there's been some literature about that. So we weren't sure whether or not inhibiting ACE2 at the transcriptional level would actually be a good idea and be beneficial. In contrast to ACE2, TMPRSS2 actually seems to be tolerated as a loss of function allele in the human population. So the PLI is 0. In knockout mice for TMPRSS2, there doesn't seem to be any severe phenotypes. The mice seem to be perfectly viable and mostly fine. And also in sort of studies looking at infection of TMPRSS2 knockout mice with SARS CoV-1, it seems like there's lower viral titers and not as big of a loss of body weight compared to just wild type mice. So it seems like TMPRSS2 could potentially be a good target for transcriptional inhibition. So we tried to do a literature-wide screen for drug treatments that could transcriptionally inhibit TMPRSS2. And the approach that we took was basically to look through the entire literature and find any studies where there were drug treatments that changed the expression of TMPRSS2 or any other gene. So we looked through Connectivity Map, which is a really well-known database of drug treatments, followed by expression profiling, as well as we looked through the entire sequence read archive. So all of GEO and all of the database where all the RNA-seq data sets are deposited. And then we also looked through some single-cell RNA-seq with drug treatments and paired gene expression values. And most of the stuff I'm going to be presenting right now is looking through the sequence read archive, where we basically just took every study that had a drug treatment within SRA, and we manually assigned whether there was a case control. We manually figured out which samples belong to a case or which samples belong to a control. We did differential expression analysis in total for about 3,000 comparisons. And we mostly did this because ACE2 and TMPRSS2 both are relatively poorly imputed by the connectivity map. And so that wasn't a really good option to look at. When we look at which drug treatments seem to significantly upregulate or downregulate TMPRSS2, we actually found that there were quite a few. And these, if you look at the preprint, these are all listed in supplementary table 1. And in supplemental tables 2 through 7, we also include regulators of ACE2, the cathepsins, and also host proteins that form protein-protein interactions with viral proteins, if anyone is interested in looking through the data. But for TMPRSS2, we find that of the treatments that downregulate TMPRSS2, 12 of the 32 treatments actually involve the estrogen or androgen pathways, involving 7 independent studies. And for treatments that upregulate TMPRSS2, they also, 24 of 76 also involve estrogen or androgen, encompassing 15 independent studies. And this matches a lot of literature, what's known in the literature of TMPRSS2 being regulated by the androgen receptor and being directly bound by the androgen receptor upstream of its promoter. And in particular, consistent with this, we find that antagonists of the androgen receptors, such as enzalutamide, which is used for prostate cancer, as well as estrogen-related compounds such as estradiol, genistein, and synthetic estrogens, all consistently downregulate TMPRSS2, while testosterone derivatives like DHT, as well as the synthetic androgen R181, seem to upregulate TMPRSS2. With regards to estrogen, we actually found a weird time-dependent effect, where you can see here that treatment with estrogen actually leads to a really short burst of increased TMPRSS2 expression, followed then by decrease in TMPRSS2. DHT, consistent with sort of what's known in the literature, leads to a really rapid increase in TMPRSS2 expression. So based on this, it seems like longer treatments with estrogen seem to decrease TMPRSS2 expression. But one of the things we were concerned about is that all the studies looking at TMPRSS2 were done in either breast cancer cell lines or prostate cancer cell lines. So all the estrogen-related studies were done in breast cancer, and all the androgen or androgen receptor antagonist studies were done in prostate cancer lines. So we tried to figure out whether or not this would actually also be relevant for lung tissue. And we have 3 different lines of evidence suggesting that this might be relevant. The first is being that the androgen receptor itself is expressed in the human lung. The second being that in castrated male mice, treatment with androgen did lead to TMPRSS2 upregulation. So we think that the converse of androgen receptor antagonists could also lead to downregulation of TMPRSS2. And finally, we looked to see in the human lung what genes were correlated with TMPRSS2 expression across different humans. And basically what we see is that there's a known estrogen response and androgen response genes are strongly correlated with TMPRSS2 expression in the lung. And then if you look to see at their dynamics of expression, you see that basically genes that are highly positively correlated with TMPRSS2 also seem to have the same expression patterns temporally following estrogen treatment, versus genes that are sort of inversely correlated have a very different expression pattern. And then for DHT treatment, you see that after DHT treatment, TMPRSS2 has a really strong upregulation, and genes really well correlated to TMPRSS2 in the lung also seem to have this upregulation, and genes inversely correlated seem to have a downregulation. So based on these 3 lines of evidence, we think that TMPRSS2 can be modulated by either estrogen or androgen-related compounds in the human lung. Because these are both like sex hormones, we also wondered if this sort of would match with epidemiological data, suggesting that sort of seeing male to female differences in susceptibility to COVID-19. But we didn't really see that much of a striking difference between TMPRSS2 expression or ACE2 expression between males and females, and also not a super strong difference in TMPRSS2 with respect to age. Although I wanted to note that there was a recent preprint that does seem to see some statistically significant differences in expression after controlling for confounders. But this is looking at lung here from GTEx, and there's not a huge difference in lung expression of TMPRSS2. But what we did notice is, especially if you look at ACE2, even though there's not a huge difference in the median expression, there is a really big difference in the variance of expression, which really surprised us, and also for TMPRSS2, where basically both the genes have a really large skew of gene expression, where most people have relatively low expressions of these 2 genes, but a small minority of people have very high expression. And if you look at the actual gene expression values, you see that TMPRSS2 in particular, the expression among 400 human individuals from their lungs, the expression varies by more than 2 orders of magnitude. So some people have only 2 transcripts per million, and some other people have 250 transcripts per million. So there's actually a really wide variability in this. And this is relatively unusual. So if you compare this against all the genes in the lung, ACE2 in particular is in the 97th percentile of skew across individuals, but even TMPRSS2 is in the 70th percentile. And we also note that both genes, especially ACE2, the skew actually increases by age. So there's less skew in younger individuals, but more skew in older individuals. And finally, the last slide is a lot of this work, the key assumption underlying the transcriptional inhibition framework is that gene expression of TMPRSS2 actually causally affects COVID-19 severity. So we just wanted to list a few of the testable hypotheses basically test this assumption. The first 2 is that you can either look at TMPRSS2 expression either before infection or during infection and sort of correlate that with with severity of COVID-19 symptoms. We note that TMPRSS2 is expressed in nasal goblet cells, and so we're hypothesizing, but we haven't tested yet, that you may be able to profile expression of TMPRSS2 from existing nasal swabs. We also note that the high skew in expression of TMPRSS2 as well as ACE2 would be consistent with a model of sort of high variability in symptoms across individuals. But of course, this is more of a hypothesis because we don't have the matched data yet for expression and phenotypes across individuals infected. But we do note that for SARS CoV, there was a previous study showing that EQTL, so genetic variants that are associated with differences in expression of TMPRSS2 across humans, was associated with severity of H1N1 and H7N9 in humans, and both of these also require TMPRSS2 entering. Also, we note that you can also use observational studies to look at estrogen and androgen and to see if people on either of those are associated with different risk for COVID-19. And in mouse, there was a paper that was mentioned a few times in the questions showing that this is the case, and also a recent preprint in humans showing that higher estrogen is associated with milder symptoms. And then finally, the best test would, of course, be a trial where you would use this either an anti-androgen in isolation or in combination to look for amelioration of symptoms. And then I just have one slide looking at with acknowledgements. So thanks to David Goldstein and the Goldstein Lab and people that helped on the study.
Thank you very much. We have a request, Michael Shen.
Yes, hi, this is Michael Shen speaking. Really interesting stuff. My lab has been interested in the hormonal regulation of TMPRSS2 with respect to this question. We're a prostate cancer lab, among other things, and as you know, TMPRSS2 is very well known in the prostate field. It was one of the first androgen-regulated genes ever discovered, and it is very well known because of its role in the TMPRSS2 ERG rearrangement, which is found in nearly 50% of prostate tumors. It's considered to be an initiating event in prostate cancer, and it also gives you an idea of how highly androgen-regulated the gene is in the prostate. And of course, women have androgens as well, so it in and of itself may just lead to some differential expression that may be significant, but maybe not readily observed at the level of overall bulk transcriptional analyses. One thing that I think is worth mentioning about TMPRSS2 is that it encodes a serine protease, but this is a secreted protein, so it can have non-cell autonomous effects. And when we're looking at expression, we have to consider the possibility that it could be acting in cell types that are, that are nearby. They're not necessarily the cells that it's expressed in.
Yeah, that's a really good point. I think especially for the prostate cancer point, given that it is fused with ERG in 50% or more of cancers, it would be really interesting to look at epidemiological data to see, if prostate cancer is associated with a reduced severity of symptoms.
Right. So right now we're starting to look at this more carefully in mouse and organoid models. To really look at hormonal regulation of TMPRSS2 in a variety of tissues and cell types, and really test ultimately whether this might affect infectivity by the virus, or by a pseudotype virus rather.
Mm-hmm. That's a really good point.
Thank you, Michael. David Salzer?
Yeah, I've been reading a paper from the Rocco group in Italy claiming that the recent COVID-2 has such increased infectivity is because it has a mutation where it's now very strongly, like fourfold more strongly binding to sialic acid residues. I wonder if you have any insight into that.
Not yet, but I haven't seen the paper yet, but I can take a look and get back to you.
Send me an email if you can, and I'll send it to you.
Yeah, that'd be great.
I have another question, Xinchen. So where did you get the RNA-seq profile for this? Are these CoV-2 infected cells, or these are the original CoV infected cells?
The RNA-seq profiles for the initial screen from the SRA, or the treatment?
Yeah, the one that you used to show that there's an increase, for instance, in the expression of TMPRSS2.
Yeah, so all the data, all the RNA-seq data that we've been using is actually just from regular prostate cancer or breast cancer cell lines. So these are non-infected, but there are some references showing that like treatment with estrogens or anti-estrogens can affect severity in mouse with SARS-CoV-1.
Thank you. Are there any other questions? Looks like Rudy, you have a question?
So I just wanted to make the observation that this, the sensitivity of this gene to gonadal steroids could account for both the very striking dichotomy in age susceptibility, that is where children don't seem to get nearly as sick from this virus as adults do, and are obviously not gonadally mature, most of them. And secondly, that it could account for obviously the gender dichotomy in adults. So this should be quite easy to test, actually, if the transcripts are available in nasal swabs, for example.
Yeah, I agree. And yeah, we're trying to move forward on the nasal swab profiling aspect now to basically test the hypothesis.
Thank you, Rudy.